Hello, my name is Imre Berger. I'm the director of the Max Planck Bristol Center at the University of Bristol, UK, and I'm delighted to give this webinar today about our discovery of a druggable pocket in the SARS-CoV-2 Spike protein and to discuss with you a new finding which we think is an avenue to defeat the current COVID-19 crisis. It is a bit unusual for me to give this presentation like this, but this kind of remote e-presentation appears to be the new normal, so we should try to enjoy this. I don't think I need to say a lot about the crisis which is currently wreaking havoc all around the globe. We have this virus, it's the SARS coronavirus 2, a new form of the SARS coronavirus, which has spread all over the world, starting from Wuhan, and it has led to major disruption everywhere. It has cost countless lives, and it is an economic pain, and we do not yet know how to get out of this crisis. And what you see here on the left side is the SARS-CoV-2. It's the coronavirus which causes the COVID-19 disease. And what you see there is basically a ball-shaped structure and with spikes coming out of it. And this is the spike glycoprotein, which also gives the coronavirus its name, because these spikes which are protruding from the surface of the coronavirus, they look like little crowns. And one of the things which we have done here in Bristol during the lockdown phase of the crisis, when we, like many others, started to work on coronavirus to try to contribute to do something about the pandemic, is to determine the structure of this spike glycoprotein trimer, which you would normally find on the surface of the coronavirus, by a technology which we use, which is called cryo-electron microscopy. And by doing so, we made a discovery which might help us to address the current crisis. Let me explain why we actually entered this coronavirus research. What happened here in Bristol was that when the crisis became more problematic, many things, almost everything that was not essential, was shutting down, including the university where I work in. At the same time, there are expert scientists here in Bristol and also elsewhere, clinicians and vaccine people, and also coronavirus researchers who would spontaneously self-organize to form groups of scientists who continued to work to investigate what this pandemic was about, to look at the virus itself, but also to develop testing methods so that one could locally test the population and to see who had actually coronavirus, who had this like COVID-19 and who didn't. And for this, they require antigens. Antigens are the proteins which are parts of the coronavirus. And my laboratory, I'm a synthetic biologist and a structural biologist, is particularly good at making proteins. So my laboratory was tasked to form a core team during the lockdown to produce antigens. And one of the major antigens, so those are those proteins which the immune system will then recognize when something foreign comes in and make antibodies against it, is exactly this spike protein, which is a trimer of 3 glycoproteins, and it's called S. So what you see on the right side is the structure which we determined as a quality control of the material which we were producing for the clinicians and for the serology people who were building up their tests. We wanted to see whether the protein which we were making was similar to what you would find on the surface of the coronavirus. And you can see on the right the cryo-electron microscopy density. So this is what we get out of our cryo-electron microscope when we look at the spike protein which we produced, and you already see that it's like a trimer. So we have one blue protein and a magenta protein and a green protein, and they look identical, and they form this trimer, which is the spike protein. So how does cryo-electron microscopy work? So basically what you do is you take a solution of the sample which you have produced, in our case the spike protein from the SARS-CoV-2 virus, and then this solution you plunge freeze. So you make vitreous out of it so that you basically get a snapshot of what is in solution when you plunge freeze it. And this frozen vitreous ice video samples in there, goes into the cryo-electron microscope. Now, all of us know how electron microscopy works, probably, because you have seen these pictures of cells, like black and white, showing like the structures within cells. So this was how electron microscopy was done years ago. In the last few years, electron microscopy has made an incredible advance, and now you can actually get not only those rough structures of what is in a cell, but you can actually get atomic structures of proteins and protein complexes by applying this flash frozen or plunge frozen cryo-electron microscopy technique. And we have like a center here in Bristol, which is headed by Christiane Schafitzel, and they are outstanding in cryo-electron microscopy. So when we produced our spike proteins for the serologists and for the other scientists who were looking at this new disease, we then also use the cryo-electron microscope to see what the structure of the proteins which we made were. That's important because you have to have the same structure which is also on the virus so that you can then confidently apply this for serology testing. And when we determine the structure, and that you do by assigning the material which comes out, that density which comes out of the cryo-electron microscope and tracing the amino acids of your protein through it, we found additional density. So we could assign everything, but then there was still density left in our structure, and we were very surprised by this. You see this here on the bottom right. So in the blue, this drawing, which represents one of the spike protein monomers, there was this extra tube of density there which did not make sense and which we could not explain by what we knew about the spike protein, also from other structures which other people have solved before. So we saw that we had some extra density there, and we did not know what this was. And the processing and the cryo-EM data collection was done by Satish and Ufuk. And what you do when you have a trimeric protein that which is kind of like where all three strands are identical, you determine the structure first in an unsymmetrized form. So there we reached roughly 3 angstrom resolution, so we could see details 3 angstrom apart. And then you apply a symmetrization where you then treat all of them equally, and that improves the data you have, and we could go to 2.8 angstrom resolution, and that's quite high resolution where you know what you see is real. So we had this extra density in there, and we knew that it was real, but we didn't know what it was. So what we did there then is first we asked ourselves that, okay, we have 3 of these individual monomers in the trimer. Do all 3 of them contain this extra density? So we looked at the unsymmetrized structure. So we made one step back, looked at the blue chain, at the magenta chain, at the green chain, and yes, in all 3 of them, we had this extra density which looked like a tube within the spike protein monomers. So all 3 sides, all 3 of these pockets which we have seen with this extra density were occupied by this extra density. We just didn't know what it was. And you see at the bottom here on the left in these 4 pictures, you see electron density all over the amino acids, also forming the pocket in which this extra density was, so that you see that the quality was really good. So you could put in the individual amino acids, and then you had this extra density. So now we had to figure out what this actually was, and this work was done by Christine, and who together with Kapil built the model into the electron density data. And what Christine did was she had a look, and she looked at this tube, which we had in there, like this unassigned extra electron microscope density. And she asked in this wonderful resource which we have, which is the protein data bank, do we see in some other structures which have been solved previously similar tubes of density which look kind of like what we had? And so she had a look there. She compared many, many structures which she found, like literally thousands of structures or tens of thousands which are in the protein data bank. And she found several proteins, including this fatty acid kinase B3, which I showed there, and also this odorant binding protein, which also had a very similar shaped tube in the proteins, and where people knew what it was, so they had assigned it properly, and it looked like it is a fatty acid, and more precisely it looked like, based on space and shape considerations and the available room in this pocket, that it and also like how the this extra density curved in this pocket that it might be linoleic acid. So linoleic acid is a fatty acid which occurs in our bodies, and it looked like what we had is actually linoleic acid based on these considerations. So we had this feeling that, okay, we have in the spike protein from the SARS-CoV-2 virus, we have extra density in there, and this extra density seems to be coming from a fatty acid which is bound, and this fatty acid apparently could be linoleic acid. So we had to nail it down, of course, and ask ourselves, what was it really linoleic acid? And what you see here on this slide is, again, the spike protein in the side view, and now also in a top view, and you see now this extra density, which is in this red box in there, which we now assigned as a linoleic acid. But at this time, it was just a proposal. So we need to find a way how to really nail it down that it is linoleic acid. And I remember that it was lockdown time, so like occasionally I was at home and you couldn't really do much except for working in the laboratory or being at home and then play badminton in the garden. And I was just moaning around about how can I find out whether this extra molecule which we have in the spike protein is really linoleic acid. And I was playing with Christiane Schafitzel, actually, who not only is a fantastic electron microscopist but also happens to be my wife. And she then said that, why don't you call Oscar? So Oscar is Oskar Stauffer, and he's a collaborator of us in the Max Planck Bristol Center. He works in the Max Planck Institute in Heidelberg, and he's a lipid expert. So he knows how to operate a mass spectrometer which can then detect lipids which are bound to our proteins. This is in Joachim Spatz's group. So Joachim Spatz is a Max Planck director who is the PI of Oscar. So I did call Oscar and I said, Oscar, we have a protein, a very interesting protein, and we think that we have a lipid or like a fatty acid in there. Could you look at it for us and find out what it is? And Oscar said, just send it. So we sent him the sample, and he put it into the extremely well-equipped mass spectroscopy the facility, and he could then find out what this extra density was, and yes, indeed, it was a fatty acid, and yes, indeed, it was linoleic acid, which you know by looking at the mass which he got out of the mass spectrometer and compare it to the chemical mass which you get from the chemical structure of the molecule. So we had linoleic acid in there. The mass was consistent with it. It is an ionization experiment, so you need to take the mass of one proton away from the calculated mass, and up to the fourth position after the decimal, the mass was exactly consistent with linoleic acid. So now we were more confident that yes, indeed, this extra molecule which we have discovered in the spike protein is a fatty acid, and it is linoleic acid, as the mass spectrometric data had confirmed. So now we could really look back again at the structure we determined and this pocket which we saw in there, and you see it now like in a magnification on the right side on this slide. So what we found was a fatty acid binding pocket specific for linoleic acid, and interestingly, and you see this in the drawing, it was bipartite. So we had like this kind of greasy tube where the hydrocarbon tail of the fatty acid was sticking in. So that's like fatty acids like linoleic acid have a hydrocarbon tail which is hydrophobic, and they have a polar head group which is hydrophilic and interacts then with positively charged amino acids or with hydrogen bond donors. So we have like this greasy part, and this was sticking in a greasy tube in one of the spike monomers, which is shown here in green. And then we had this other part which was kind of like the polar lid interacting with the polar head group of the linoleic acid, and that you see here in green. So there were actually two of the monomers involved, and not just any part of the monomers, but a very special part of the spike protein is where this pocket is in there. It's that part which is the so-called receptor binding domain. So that domain which then attaches to a protein, as we will see in a moment, on the surface of cells, then and leads to the infection and to the disease when you get unlucky. But apart from that, just based on like structural considerations, when we looked at this pocket, and you see this now at the bottom in a 3D representation showing the surface charge and the hydrophobicity pattern in this pocket. So this is the kind of pockets which people who are in drug discovery really linoleic because it's a pocket which very smugly fits around this linoleic acid. So you can design a different small molecule which fits in there and will possibly displace this linoleic acid, and that is very, very important, as you will see in a moment. And when we had all this data together, we wrote this up, and this is also the reason for this talk, for the invitation by GeneScript to give this webinar. So we wrote this manuscript together. We found an unexpected free fatty acid binding pocket in the cryo-EM structure of the SARS-CoV-2 Spike protein, and the manuscript is currently under review and available as a preprint on bioarchives, because during this crisis, more than any time else, one shares all the data one has to inspire other people to find possibly ways to deal with the COVID-19 disease or with the virus, and that's why you deposit your new findings on this freely accessible public preprint server, which is bioarchives. All right, so here we were, and we had discovered this binding pocket, and the binding pocket was bipartite, so very interesting, different than normally, because normally, like linoleic acid binding proteins would contain the lid and the greasy tube in one protein, but we had two proteins contributing to this. So this was all very interesting, but you know, as a structural biologist and generally as a scientist, you have to always be very careful whether that what you see there is real, and whether if you see something which nobody else had seen before, whether you are still dealing with a protein which is actually functional and you didn't introduce something in there due to your experiment which doesn't represent reality. So we needed to find out whether the spike protein with this linoleic acid bond, which we had discovered, whether this would be still functional. And this was now again an issue, that how do you do this? So during this crisis, what really impressed me was that, you know, the problems were coming, but the solutions were also coming very quickly after each other. So not like 2 or 3 months normally as it would take, but you have a problem, but the solution comes like somehow also very, very fast. And so when we had this situation that we needed to check the functionality of this spike protein which we had made, and we didn't know exactly how, then my good friend Tiro from GeneScript, I remember this, he wrote me an email in the middle of the night, roughly around midnight, where he had said that like they came across like a new surrogate virus neutralization assay, which sort of allows you to test samples and also serum and antibodies and whatever you have which might be relevant to COVID and to SARS-CoV-2, which can let you test this in your laboratory in a normal laboratory safety environment, so where you don't have to work with tissue, where you don't have to work with human samples and with live virus even, and go then into the category 2 and category 3 labs, which we didn't have any access to. So when I was thinking about like how can I show the functionality of the spike protein we made, then we got this email from Tiro providing the solution. And it was the surrogate virus neutralization kit, which is now called CPAS Smart Kit, I think, in GeneScript. And it works very cleverly. So how this works is that normally on human cells you would have this receptor which the virus binds to, with the coronavirus when it comes in, and that's called ACE2. So it's like a protein called ACE2 that sits on the surface of the cells. So in this assay, what they did was to deposit the same protein, ACE2, on a microtiter plate on the surface. So now you have the ACE2 sitting as it would normally sit on the surface of the cell. You have it sitting on a microtiter plate. And added a piece of the spike protein, exactly this receptor binding domain, and put a color reagent on it that's called horseradish peroxidase. So when you add a chemical, it produces a color. So what happens now is that if you add this RBD, this receptor binding domain, this isolated recombinant receptor binding domain with that color reagent on it, to your plate, then it will bind to the ACE2 and it will produce a color. Now, when you titrate in either virus or spike protein or serum from patients into this reaction, you will displace the RBDs which are bound there and you will reduce the color. So you will know that your material is functional because it still binds ACE2 by looking at the color disappearing in the assay. And that's exactly what we did. So we could show that our spike protein with the LA bond would very competently compete away the RBD with the HRP, and we would lose the color, and we could even determine how well our spike protein bound to ACE2. So now we knew that it was functional, and we also confirmed this by a size exclusion chromatogram, where we mixed the ACE2 protein and the spike protein and then ran it over a column and saw that they were coming out together. So we knew now that the spike protein with the LA in there was functional as people had described before when they didn't know that it had LA bound, and it would bind ACE2. And how this works is you see on a model on the right. So we have in blue again this receptor binding domain of the spike protein, now with our LA sitting there in the pocket. In red I have colored the amino acids which are involved in the binding of ACE2. ACE2 is showing here in a yellow, and you see that there's a very tight complex between this red part, which is called Achilles heel, because that's the sensitive part of the spike protein, which it needs to react with the cellular ACE2 receptor to get into the cells. That is bound by ACE2, and this is how this works, and our material was functional. So thank you so much, Tiro, for helping us out in there by providing early access to a better version of the kit which you were putting together. So we now knew that our material had LA in there and that it was functional, so our spike protein was functional. So now we could ask ourselves, what are the structural features that define this LA binding? And we could find 4 features which I show here. So one is that we have this greasy tube into which the hydrocarbon tail of the linoleic acid fits in there. And we have at the beginning of this greasy tube, we have a gating helix. So that's like a helix which needs to move away so that the LA can penetrate into there. And this we could find out by comparing the apo structures people have determined before when they didn't know yet that it was LA binding, and our structure with the LA binding itself and superimposing them. So we saw the gating helix. The gating helix has big aromatic residues like tyrosines which swing open like a door when you're pushing the gating helix away, and when the linoleic acid can then penetrate into this greasy tube. We also have a polar lid, as I said before, coming from the next receptor binding domain, and that has consequences because you have like this next receptor binding domain which shuts the lid on the linoleic acid when it has penetrated into there by interacting with the polar head group of the fatty acid, and this brings the RBDs together. So what you have there, you have a situation, and this will become very important, that the receptor binding domains, which normally would be loosely attached and with space in between them, are coming close together and end up in a condensed form. So those are the 4 architectural features which define LA binding by the coronavirus spike protein. And now we could, of course, ask ourselves that how conserved are those features? There are currently 7 coronaviruses which we know. 4 of them are circulating. They are like harmless coronaviruses. They have jumped from animals already earlier over to us in humans, and they are the common cold. They are not lethal. And we have 3 very lethal coronaviruses. That's the current SARS-CoV-2, which causes COVID-19. But we had, of course, before, like 15 years ago, also SARS-CoV, the original SARS coronavirus, which was extremely lethal, but not that infectious as what we have now. And we had also the MERS, like the Middle Eastern respiratory syndrome coronavirus, which is also very lethal, but was luckily also not that infectious. So we could now have a look and ask, is this fatty acid binding pocket conserved, and is this linoleic acid binding maybe somehow an element of pathology of the viruses? And to cut a long story short, what we found is that exactly those elements which we found in SARS-CoV-2 are conserved in SARS-CoV, so very lethal other coronavirus. And even though the MERS-CoV has separated thousands of years ago from SARS, and is in the Middle East and not in China where the SARS coronavirus comes from, it also has the features of which we have seen in SARS-CoV-2. So it could be, and it's very possible, that this linoleic acid binding is an element of pathology. So why are we thinking that that could be the case? So what is so special about this linoleic acid? So now when we had the data together and we saw that in the other pathogenic coronaviruses, this linoleic acid binding pocket is apparently also conserved, we looked very deeply into what is linoleic acid actually. And it turns out linoleic acid is an incredibly exciting molecule. It is an essential free fatty acid, so it cannot be synthesized in cells, you have to take it up by the diet. And linoleic acid is an important molecule, a precursor for a metabolic pathway with which the body then synthesizes out of this essential free fatty acid arachidonic acid and eicosanoids, including prostaglandin. And those are molecules which are central to inflammation and immune modulation. And those are key symptoms of COVID-19. So this systemic inflammation everywhere in the body is what kills people when they get the disease. And you can already see that if you do something like if you scavenge this linoleic acid out of this metabolic pathway, that you will have like detrimental effects on those molecules which would protect you against inflammation and other symptoms of COVID-19. Linoleic acid is also essential for maintaining membrane fluidity because it's an unsaturated fatty acid, and that's what makes your membranes of your cells fluid. And this, again, is important for the surface tension in lungs, where the air has to penetrate through the membrane into the cell so that the oxygen can be taken up. And it has been shown in different diseases before that if you mess with the linoleic acid pathway lipid composition, it will lead to acute respiratory distress syndrome and severe pneumonia, which are both, again, key elements of COVID-19 pathology. And a third aspect which struck us was that linoleic acid directly binds to a receptor, free fatty acid receptor, which is called FFR1, GPR40. And this receptor is a key regulator in insulin disease, which is central to diabetes, as you all know. And diabetes is a risk factor for COVID-19 patients. So people who have diabetes die much more frequently from the disease than people who do not have diabetes. So, and they already have an impairment in the LA signaling, which is exacerbated by the virus. And what came as like an incredibly interesting finding very recently was a paper in Cell, where people described the lipidomic and proteomic situation in the sera of COVID-19 patients. And you can see there that the LA levels are continuously decreased. So altogether, if you now take this together and digest it, then you realize that our discovery provides the first direct structural link between linoleic acid, COVID-19 pathology, and the SARS-CoV-2 virus itself, because the spike protein binds directly to this linoleic acid, which is so central to all these processes which have repercussions on disease development in COVID-19. And our results suggest that both the LA binding pocket within the S protein and the multinodal LA signaling axis involved in all kinds of metabolic pathways and inflammation and immune modulation represent excellent therapeutic intervention points against SARS-CoV-2 infections, particularly in patient groups which have increased risk due to metabolic preconditions like people who have diabetes. So this is our discovery. So now, okay, so we have this pocket, we have linoleic acid in there, we know that it's like potentially an element of pathology. It is conserved in SARS-CoV and also in MERS-CoV. So what can we do about this? Is there like a technical way to now proceed and use this information which we have to develop an approach to defeat COVID-19? So let me now come back again to the spike protein itself. So I told you that when we have cryo-EM, we have a snapshot of what is in solution. And when you look at what you have on the cryo-EM grid, you can look at the individual molecules. And you see that the spike protein exists in two forms. So there's this open form, which you see here on the left, where you can see that this blue protein, the receptor binding domain, is sticking out. And in red, again, we see the Achilles heel domain, and this is ready to engage with the ACE2 receptor and then to penetrate into the cells and trigger those events which cause the infection and the disease if you are unlucky. So this form is called the open form, which is competent in receptor binding, which is the infectious form. On the other hand, you have this closed form. So in this closed form, this receptor binding domains, all three are tuckered in, as you can see here, and these red Achilles heel domains are hidden and cloaked away. So why does the virus do this? So this is the closed form. Here in this closed form, the Achilles heel domain of the receptor binding part is hidden, and this form is not infectious because it cannot bind the receptor. And why does the virus do this? This is a way of avoiding the immune surveillance. So when the virus comes into the body, it has to avoid being targeted by antibodies which the body produces, which would eliminate infectivity. So often it tries to hide that critical part, the Achilles heel, with which it would bind the receptor, but it has to swing out sufficiently often that when an ACE2 receptor is there, it can latch onto it and it can get into the cells and cause infection. So what you have is a dynamic equilibrium. You have the open form, you have the closed form. The open form is ACE2 competent, binding competent, and it's infectious. The closed form cannot bind the receptor, so it's not infectious. And now what we saw when we looked at that is that in our case, when we just counted the number of open and closed forms on the grid, so in this snapshot of what was in solution, we had 30% of the spike proteins were in the open form, and 70%, so like the majority, was in the closed form. So in our case, the not infectious form had a preponderance. In the structures which were determined before us, which didn't have LA bound, where people did not have LA in there, the open form, so the infectious form, was dominating. So you had more of the infectious form and less of the closed non-infectious form. So this already told us that you can influence the relative amounts of infectious virus and non-infectious virus, for example, by LA. And how does this work? So when we now look in a schematic representation, I'm trying to show this now schematically. Here we have in the middle, we have the structure with the three linoleic acids in there, and again, like blue and green and magenta are the three individual spike proteins. And on the left, I have shown you how the APO form without LA looks like by looking at the structures which people have done before. So you have this loosely arranged receptor binding domains, and you have these canyons in between. So you can imagine how these can quite nicely swing out and then contact the ACE2 receptor. In our case, however, the LA, because it binds two RBDs at the same time, acts as a staple. So it's tuckering down the receptor binding domains, and they have a harder time to swing out. So it's not completely gone yet, but the equilibrium is shifted towards the non-infectious form because the LA sits there and it tuckers the two RBDs each together, and it makes this closed, locked down form which we observed in our structure. Could this be used to develop now something, an approach, an antiviral, which would always keep the spike protein in the locked conformation? So we looked in the literature, and yes, there is a precedent for this. It was done by Michael Rossman himself, who is a titan in structural virology, and he was drugging rhinovirus and defeating rhinovirus using exactly the same concept which we are now proposing for the SARS-CoV-2 coronavirus, which causes COVID-19. So rhinovirus causes the common cold, so it's not such a major disease, but still for people who have asthma, very dangerous. So what they found is that in the rhinovirus, you have these viral proteins, which are shown schematically here in blue. You have a canyon between them, again, and you have a pocket where an unidentified fatty acid binds. So they couldn't identify it, but normally there would be a fatty acid binding in that. So what they now developed are drugs which displace the fatty acid, go in there, and then lock this viral protein in a form which can no longer be recognized by the receptor. So the receptor has to be, the receptor, as you can see on the left, binds a very defined structure which is caused by the fatty acid binding there and the viral protein sitting on top of it. And when you now put a bulky chemical in there which binds better to that pocket than the fatty acid, and you distort this viral protein and you sterically block it in a form that it cannot bind the receptor anymore, then you abolish infectivity, and this was exactly what happened with rhinovirus. So based on this, you developed small molecules, and two of them are currently in the clinic, also as inhalable molecules, and it's a successful drug development to shut down the infectivity of coronavirus. So what do we now want to do? I think it's entirely obvious. So we have already learned that without linoleic acid, we have the majority of the proteins, the SARS-CoV-2 S proteins in the open form, which is infectious competent. When we add linoleic acid, then we shift the equilibrium towards a form which is closed predominantly and less infectious. Now, if we can develop like Rossmann did, a super linoleic acid mimic antiviral, which would always shut down the spike protein in the closed conformation, we could eliminate SARS-CoV-2 infectivity and therefore shut the virus down before it even enters the cells. And this is what we would like to do now. So let me recap our findings. So we found a linoleic acid binding pocket in SARS-CoV-2 Spike and defined the characteristics of it at near atomic resolution by cryo-electron microscopy. We found, based on structural considerations, that this is likely conserved in highly pathogenic SARS-CoV, the original SARS virus, and MERS-CoV coronaviruses. The pocket is bipartite, so it connects two receptor binding domains, each within the S trimer. It stabilizes upon LA binding the closed form of spike with the RBDs locked down and tucked in, and this closed form is not or less infectious. There is a precedent, rhinovirus, as I have shown you, which conveys a small molecule antiviral strategy. And you have heard a lot of the hype which is going on now about vaccines against coronavirus. But remember that in the case of HIV, which is another historic precedent, after 30 years of intensive research, we still do not have an HIV vaccine. And what works are antivirus, a cocktail of small molecules which keeps the virus at bay, and this encourages us because it means that the same could be possibly done with SARS-CoV-2, the virus which causes COVID-19 disease. I want to thank at this point the great people who volunteered to do this work, because when the university locked down, then continuous working was only on a voluntary basis, and I had these fantastic people who volunteered and who wanted to continue working, to contribute, to do what they could to contribute to defeat the crisis or to do something about it. And I, of course, also thank the many funders who are funding my lab, because we are using that funds which we got from them now to do and fuel our COVID research. I would in particular thank GeneScript and Oracle. Oracle was fantastic. They provided the cloud computing infrastructure which we needed to calculate our cryo-EM structures and process that data at the speed which was required. And I want to mention where this work happened. It was in the Max Planck Bristol Center. It's a fantastic collaboration between the University of Bristol and the Max Planck Gesellschaft in Germany. And this center was critical to identify linoleic acid because we had access to the mass spec equipment and expertise of our collaborators at the Max Planck Institute in Heidelberg. There is a lot of other work which I couldn't allude to today taking place at the Max Planck Center, not only in the context of COVID-19, but in the context of COVID-19, we are developing there also synthetic vaccines, where we hope that we can elicit a strong immune response in case that the vaccines which are currently being developed do not work so well. And we are also working on synthetic mega antibodies as passive immunization reagents for people who are very sick and who cannot make antibodies themselves. And with this, I would like to thank you for your attention and come to an end. And I think it's obvious what we want to do now. Based on our groundbreaking discovery, we want to develop small molecule antivirus, as Michael Rossmann did, to abolish SARS-CoV-2 infectivity. I thank you for your attention.